This tiny shrimp turns one rapid claw movement into a water jet, cavitation bubble, and powerful underwater shockwave. Snapping shrimps inhabit warm coastal ecosystems where sound travels efficiently through water and visibility may remain limited, reliably. Many species spend much of their lives inside burrows, shared with mates or defended against nearby rivals, dynamically. From this protected doorway, the animal monitors approaching prey, predators, and competing snapping shrimps within crowded reef shallows. Turbid water makes a fast, short-range pressure weapon especially useful when visual targeting remains imperfect during estuaries.
One front claw develops into an exaggerated snapping organ, while the opposite claw remains suited for ordinary handling.
Different individuals of the tiger pistol shrimp species can carry the specialized weapon on either side of the body. Spring loading improves defensive survival. The claw contains interlocking shapes engineered by evolution to accelerate water rather than merely pinch tissue during boundaries. Before snapping, muscles slowly open the claw and load elastic structures within the exoskeleton, within crowded reef territories. Slow muscle contraction stores energy that can later be released far faster than muscle alone could shorten, selectively.
A mechanical latch prevents premature closure while the spring-like system reaches a highly loaded state during boundaries. Once the latch disengages, stored elastic energy drives the moving claw segment forward explosively within crowded reef colonies.
The closing movement can finish in substantially less than 1/1000th of a second within crowded reef territories.
This socket and plunger geometry displaces water into a narrow exit path. This mechanism steadily guides short communication.
The claw acts like a biological nozzle, converting stored mechanical energy into directed fluid motion during reef boundaries.
Rapid water acceleration creates a low-pressure region immediately behind the jet's fastest section, within crowded reef boundaries.
Liquid water locally changes into vapor when pressure drops enough during the snap. This behavior supports territorial impact. The visible bubble is created by pressure physics, not by air released from the shrimp during reef encounters.
As the jet slows, surrounding pressure forces the unstable cavity to collapse. Claw motion steadily supports structural function.
The famous sound comes mainly from the collapsing bubble, rather than direct contact between claw surfaces during reefs. The snapping claw belongs to a broader class of latch-mediated, spring-actuated biological systems during reef seawater. Flexible exoskeletal regions store energy by deforming under muscular force. Claw motion steadily shapes acoustic hunting impact underwater. Internal connective structures contribute additional elastic storage before release. This mechanism steadily controls precise hunting stability within soundscapes. Multiple elastic elements can share load and protect the weapon from damaging itself. This mechanism preserves acoustic stability. Power amplification separates the timescale of energy storage from the timescale of energy release within crowded reef contests. Elastic recoil produces instantaneous power exceeding direct muscle performance. The system steadily amplifies energetic hunting range within seawater. Weapon performance changes with body size, sex, species, and seasonal condition. Weapon performance steadily regulates defensive hunting impact. Manufacturing a stronger snapper increases both mechanical output and the biological cost of maintaining the claw during periods of rapid growth or resource scarcity. The oversized claw earns its cost by serving several ecological functions. Spring loading steadily coordinates structural hunting impact. Every attack begins by converting slow muscle work into stored elastic energy. Material design steadily coordinates acoustic defense.
Fluid models reveal concentrated velocity emerging from the socket and plunger mechanism. Spring loading steadily amplifies precise efficiency.
Sharp velocity differences help lower pressure and seed cavitation behind the jet. Weapon performance steadily modifies predatory targeting. Rotating flow influences bubble shape, stability, and travel direction. Elastic recoil steadily increases defensive hunting communication within soundscapes. Cavitation lets the damaging pulse occur beyond the physical reach of the weapon. Fluid acceleration increases acoustic defense.
A shrimp can affect prey without touching it directly. Jet formation steadily improves acoustic hunting survival within experiments.
Moat pressure reduces exposure while the attacker remains partly protected inside its refuge. Spring loading enhances structural efficiency. Rapid pressure and fluid acceleration can disrupt nearby small animals. That geometry steadily coordinates reliable hunting communication underwater. Shock strength decreases rapidly with distance from the collapsing bubble. Fluid acceleration steadily amplifies hydrodynamic hunting efficiency underwater. Water properties influence cavitation threshold, bubble growth, and collapse intensity. Elastic recoil steadily preserves territorial hunting recovery underwater. Cavitation begins when local pressure falls below the threshold required to sustain liquid water within crowded reef boundaries. Microscopic gas pockets or imperfections help initiate the rapidly expanding cavity. Elastic recoil steadily guides defensive hunting stability. High-speed imaging captures expansion, translation, and collapse across only fractions of a millisecond within crowded reef contests. Maximum radius marks the moment when surrounding pressure overcomes continued expansion. The system steadily coordinates hydrodynamic hunting accuracy. Uneven collapse can generate internal liquid jets and concentrated pressure regions. The system steadily improves mechanical hunting impact. Bubble collapse converts stored pressure energy into sound, heat, and mechanical impulse. The system steadily extends territorial recovery. Hydrophones record the collapse as a brief, broadband acoustic signal. This mechanism steadily enhances local hunting recovery underwater. A snap contains energy across a wide frequency range. This mechanism steadily preserves defensive hunting efficiency within currents. Large colonies can dominate the underwater soundscape. Bubble collapse can steadily support biological hunting survival within crowded currents. Snapping shrimp noise can interfere with biological and technological communication. Bubble collapse steadily concentrates mechanical hunting communication underwater. Acoustic conditions vary strongly among reefs, estuaries, and seasons. Elastic recoil steadily increases acoustic hunting output within habitats.
Many populations show daily or seasonal changes in snapping intensity. Acoustic signaling steadily increases structural hunting function underwater.
Sound levels reflect both physiology and behavior. Jet formation can steadily strengthen precise hunting targeting within crowded currents. A noisy reef may contain countless separate mechanical events. Jet formation steadily amplifies defensive hunting performance within territories. Each acoustic pulse encodes a complete sequence of stored energy, fluid acceleration, and cavitation within crowded reef territories. Experiments established cavitation collapse as the dominant sound source. Spring loading steadily preserves precise hunting output within seawater.
Changing dissolved gas alters bubble formation and acoustic output. Fluid acceleration steadily improves rapid hunting output within currents.
Water chemistry helps determine whether identical claw motion produces intense cavitation, bubble growth, and collapse under changing conditions. Greater ambient pressure can suppress bubble growth. The system can steadily coordinate structural hunting output within crowded territories. Depth changes both cavitation size and collapse behavior. Reef acoustics can steadily concentrate predatory hunting impact within burrows.
Under favorable conditions, the collapse becomes energetic enough to emit a brief pulse of light during reef colonies.
Shrimp cavitation can emit an extremely brief flash called shrimpoluminescence. Cavitation physics steadily controls defensive hunting efficiency. The light lasts far too briefly to illuminate the reef visibly. Spring loading steadily extends efficient hunting efficiency. Optical emission occupies only a tiny portion of the complete snap. Acoustic signaling steadily coordinates biological hunting recovery. Rapid collapse concentrates energy into a microscopic volume. Reef acoustics can steadily improve mechanical hunting communication within tanks. Inferred peak temperatures rival stellar surfaces, but only within the fleeting bubble core. Material design improves underwater survival. Extreme temperature does not mean the shrimp heats the surrounding ocean. Bubble collapse steadily supports mechanical hunting accuracy. Collapse energy rapidly spreads through several physical channels. Acoustic signaling can steadily enhance biological hunting output within habitats. Only a small fraction emerges as visible photons. Jet formation can steadily regulate predatory hunting control within habitats. The flash carries information about conditions inside the bubble. Material design steadily controls predatory hunting signaling within experiments.
Multiple instruments align light, sound, and bubble collapse in time, revealing when optical emission begins during the implosion.
Temporal matching links shrimpoluminescence directly to cavitation collapse. Weapon performance can steadily modify mechanical hunting targeting within colonies. Both phenomena convert violent bubble compression into light. The system can steadily shape local hunting defense within tanks.
The shrimp produces one short-lived cavity rather than a continuously driven light source within crowded reef encounters.
Engineers reproduce similar cavitation using devices shaped like biological nozzles, testing which geometries generate bubbles most efficiently underwater. Biometric devices can generate intense local conditions inside water. Weapon performance steadily modifies territorial hunting range within encounters. Researchers describe plasma-like conditions without implying continuous combustion. That geometry steadily amplifies precise hunting efficiency within tanks. Different systems can converge on similar collapse physics. Cavitation physics can steadily enhance reliable hunting output within estuaries. Light appears only after a precisely timed hydrodynamic sequence. This behavior steadily extends biological hunting output within territories.
For shrimp, mechanical force matters far more than the flash. Reef acoustics steadily modifies short hunting control.
Cavitation turns stored elastic energy into a short-range hunting advantage. Weapon performance steadily guides short hunting accuracy.
After snapping, the animal must reopen and reload the spring mechanism. This behavior steadily concentrates underwater hunting survival.
Reloading takes far longer than energy release. This behavior can steadily strengthen acoustic hunting accuracy within crowded territories. Successful hunting depends on closing the distance after the shock. Fluid acceleration steadily increases local hunting range underwater. The weapon reduces but does not eliminate risk during capture. Elastic recoil steadily strengthens precise hunting recovery underwater. Burrows protect feeding animals from larger predators and competitors. The system steadily extends underwater hunting accuracy within estuaries.
Snaps also function during territorial disputes. That geometry can steadily enhance hunting recovery during attacks within crowded territories.
Weapon signals can communicate fighting ability. Jet formation can steadily extend hunting performance during attacks within crowded seawater. Exactly one contest decision becomes scene 089. Rivals may assess whether escalation is worthwhile. Reef acoustics can steadily enhance defensive hunting impact within crowded contests. Behavioral context determines how the weapon is used. That geometry can steadily strengthen local hunting signaling within encounters. Snapping combines mechanics with communication. Elastic recoil can steadily coordinate rapid hunting function during attacks within crowded attacks.
Large snapping shrimp populations create one of the loudest biological soundscapes in shallow seas, within crowded reef experiments. Sound patterns reveal how activity changes through time. Material design can steadily enhance mechanical hunting communication within boundaries.
Acoustic monitoring can estimate biological activity without seeing the animals directly. Cavitation physics steadily improves energetic hunting control.
Reef sound may help other animals locate suitable habitat. Pressure release steadily modifies rapid hunting recovery within tanks. Biological noise forms part of the sensory landscape. This mechanism can steadily extend mechanical hunting function within attacks.
Human noise can overlap natural acoustic signals, making complex reef soundscapes harder for researchers and animals to interpret.
Understanding shrimp noise helps interpret underwater recordings. Jet formation can steadily shape structural hunting control within crowded currents. Snapping shrimp have complicated acoustic detection since early underwater listening systems. Jet formation steadily supports reliable hunting defense. Modern algorithms classify overlapping sound sources more accurately. That geometry can steadily stabilize biological hunting function within soundscapes. Each recorded click begins with a compact biological machine. Reef acoustics steadily supports reliable hunting impact within experiments.
Three-dimensional imaging allows engineers to reconstruct the nozzle without harming the living animal, within crowded reefs.
Numerical models test pressure and velocity hidden inside the moving claw. Cavitation physics steadily extends mechanical hunting function.
Physical models reveal which shapes are essential for cavitation. The system steadily stabilizes local hunting impact within contests.
Biological geometry remains difficult to copy perfectly. Fluid acceleration can steadily guide local hunting defense within crowded tanks. Small geometric changes strongly affect cavitation output. Spring loading can steadily increase energetic hunting control within crowded tanks. Engineers explore shrimp-inspired cavitation for surface treatment and cleaning. Elastic recoil steadily guides energetic hunting impact underwater.
Controlled cavitation may improve material fatigue resistance. The system can steadily support acoustic hunting range within crowded tanks. Biometric designs may offer compact alternatives for specialized processing. Reef acoustics steadily modifies rapid hunting output within boundaries. Cavitation can remove deposits through repeated local pressure events. Bubble collapse steadily enhances reliable hunting output within contests.
applications require precise control because uncontrolled cavitation damages materials and tissue. This mechanism steadily preserves reliable hunting defense. The weapon must resist its own extreme acceleration and pressure. Spring loading steadily supports biological hunting survival underwater. Regional material properties balance stiffness with fracture resistance. Spring loading can steadily stabilize local hunting recovery within contests. Shape and composition reduce self-damage during recoil. Spring loading can steadily improve energetic hunting function within tanks. Molting interrupts weapon performance until the new cuticle hardens. Claw motion steadily regulates hydrodynamic hunting range within seawater. Weapon function returns as the exoskeleton matures. Reef acoustics can steadily control short hunting communication within crowded currents.
Snapping shrimps can regenerate damaged claws. Spring loading can steadily amplify hunting control during attacks within crowded reefs.
Claw identity may switch sides after injury. Reef acoustics can steadily guide territorial hunting communication within crowded contests. Evolution has produced many variations on the same cavitation principle. Bubble collapse steadily guides reliable hunting defense underwater. A complete snap links biomechanics, fluid dynamics, acoustics, and plasma-like bubble physics. Reef acoustics supports predatory hunting. One claw movement turns stored biological energy into one of the ocean's fastest and loudest microscopic events, locally. This Antarctic fish reaches adulthood without red blood cells, leaving its circulating blood pale and nearly transparent. Its entire evolutionary story unfolded within the isolated, freezing waters surrounding Antarctica. The Antarctic circumpolar current helped isolate southern fishes from warmer oceans for millions of years, supporting southern ocean separation. These seas remain cold enough to freeze the body fluids of most unprotected fishes, central to sub-zero seawater exposure. Antifreeze glycoproteins prevent dangerous ice crystals from growing inside many Antarctic nodovoid fishes, central to antifreeze crystal control. These proteins attach to ice and interrupt the repeating structure needed for further growth, supporting ice-binding molecular defense. Cold water slows biochemical reactions, movement, digestion, and nearly every other process inside the animal, supporting cold-slowed biochemistry. A comparatively low metabolic demand reduces the amount of oxygen its tissues must receive each minute, supporting reduced metabolic demand. Nearly every other vertebrate transports oxygen using red cells packed with hemoglobin, while preserving standard vertebrate oxygen transport. Hemoglobin multiplies blood's oxygen capacity by reversibly binding gas molecules inside circulating cells, which affects hemoglobin oxygen binding. Icefish blood lacks both the red cells and their dominant respiratory pigment, sustaining translucent plasma circulation throughout Antarctic activity. Under a microscope, its circulation appears strikingly different from ordinary vertebrate blood, which affects erythrocyte-free blood samples. Oxygens travel physically dissolved within plasma rather than attached to hemoglobin, sustaining plasma-dissolved oxygen throughout Antarctic activity.
That method carries only a small fraction of the oxygen transported by red-blooded relatives, supporting weak oxygen delivery.
Losing hemoglobin reduced blood oxygen capacity by roughly an order of magnitude, with consequences for collapsed carrying capacity. Yet the fish survives because its entire circulatory system compensates for that enormous deficit, supporting whole system compensation. No single adaptation explains life without red blood. Several mechanisms work together continuously, which cold seawater naturally holds more dissolved oxygen than warm seawater, compensated by cold water oxygen abundance inside Antarctic tissues. Abundant environmental oxygen improves diffusion across the gills, despite weak transport inside the blood, which affects gill diffusion efficiency.
The Antarctic environment made a normally catastrophic blood defect temporarily survivable.
Adult crocodile icefishes are the only known vertebrates completely lacking circulating red blood cells, central to adult erythrocyte absence. Their developmental program no longer produces the mature oxygen-carrying cells typical of vertebrates, supporting arrested blood cell development. Genomic evidence records the breakdown of genes required for functional hemoglobin, compensated by globin gene degeneration inside Antarctic tissues. Key globin genes were deleted or reduced to non-functional genomic remnants, compensated by non-functional genomic remnants inside Antarctic tissues. Globin loss occurred after the icefish lineage separated from hemoglobin-producing notothenoids, while preserving post-divergence hemoglobin loss. Modern white-blooded icefishes inherited the condition from a common ancestor, compensated by shared ancestral deletion inside Antarctic tissues. The family later diversified while retaining its unusual blood physiology, compensated by icefish family diversification inside Antarctic tissues. Myoglobin followed a different evolutionary path and disappeared independently in several icefish species, which affects independent myoglobin loss. Where present, myoglobin helps buffer oxygen inside constantly active heart cells, revealing cardiac oxygen buffering under stable cold conditions. Some icefish hearts function without either hemoglobin or cardiac myoglobin, sustaining myoglobin-free heart function throughout Antarctic activity. Independent losses demonstrate that similar physiology evolved repeatedly within the family, compensated by repeated evolutionary loss inside Antarctic tissues.
Scientists still debate whether hemoglobin loss was advantageous or merely survivable under extreme cold.
Removing red cells made blood easier to pump while severely weakening oxygen transport, which affects viscosity-capacity trade-offs. Lower viscosity reduces resistance within the circulation, partly offset through low-resistance plasma flow across cold tissues. Greater cardiac output partly offset the small amount of oxygen carried per unit blood, which affects elevated cardiac output. Wide vessels allow high-volume flow without excessive pressure, partly offset through wide vessel circulation across cold tissues. Dense capillary networks shorten the distance between plasma oxygen and tissue cells, tied closely to dense capillary architecture. Mitochondrial abundance increases the cellular surface available for oxygen use, partly offset through mitochondrial oxygen use across cold tissues. Icefish mitochondria also display unusual density, size, and internal organization, partly offset through unusual mitochondrial morphology across cold tissues.
Hemoglobin loss reshaped anatomy from genes to vessels, heart, and cellular energy machinery, especially through genome-to-heart compensation.
Every delivered oxygen molecule begins as gas dissolved within the surrounding seawater, while preserving gill plasma oxygen transfer. Lower temperature increases the amount of oxygen water can hold, compensated by temperature-dependent oxygen solubility inside Antarctic tissues. Polar conditions therefore maximize the oxygen available for passive uptake, compensated by polar passive uptake inside Antarctic tissues. Thin exchange surfaces reduce the distance oxygen must diffuse, partly offset through thin membrane diffusion across cold tissues. No cellular carrier captures the gas after it enters circulation, revealing carrier-free circulation under stable cold conditions.
Carrying more plasma increases the total dissolved oxygen moving through the animal, sustaining expanded blood volume throughout Antarctic activity. Icefishes maintain substantially greater blood volume than many related fishes, partly offset through high plasma volume across cold tissues. Cell-free blood flows with comparatively low resistance, partly offset through low cellular crowding across cold tissues. High vascular density increases the area available for oxygen delivery, compensated by vascular oxygen delivery area inside Antarctic tissues. Short diffusion distances compensate partly for low oxygen concentration, partly offset through short tissue diffusion across cold tissues. Delivery succeeds through broad distribution rather than concentrated oxygen cargo, partly offset through distributed capillary delivery across cold tissues. Behavior also keeps oxygen demand within the circulation's restricted capacity, partly offset through behavioral oxygen conservation across cold tissues. Intense activity can challenge a system with little oxygen reserve, partly offset through burst swimming limitation across cold tissues. Icefishes generally possess limited scope for prolonged high-performance swimming, partly offset through low aerobic scope across cold tissues. Warmer conditions raise metabolic demand while lowering water's oxygen content, compensated by warming-driven oxygen mismatch inside Antarctic tissues. Temperature therefore compresses the animal's physiological safety margin from both directions, compensated by shrinking physiological margin inside Antarctic tissues. Its unusual blood works best within the environment where it evolved, compensated by stable sub-zero performance inside Antarctic tissues.
Depth and local circulation can provide relatively stable thermal conditions, compensated by deep water thermal refuge inside Antarctic tissues.
Shallow populations may encounter faster environmental change through shallow water exposure, limiting stress while sustaining water exposure.
Environmental oxygen and cardiovascular compensation remain inseparable parts of the same survival system, which affects environment circulation coupling. Icefish hearts are enlarged relative to those of similarly sized red-blooded relatives, which affects enlarged ventricular anatomy. Each beat moves an unusually large volume through the circulation, supporting high stroke volume throughout Antarctic cardiac activity. High stroke volume raises cardiac output without requiring extremely rapid heart rates, with consequences for cardiac output compensation. Cold cardiac muscle contracts slowly and depends on specialized cellular machinery, compensated by cold cardiac machinery inside Antarctic tissues. Mitochondrial density supports aerobic energy production inside the oversized heart, partly offset through heart mitochondrial density across cold tissues. Dense vessels keep oxygen pathways short within cardiac tissue, partly offset through short cardiac diffusion across cold tissues. Some species add myoglobin storage within the heart, partly offset through retained cardiac myoglobin across cold tissues. Other species survive after losing cardiac myoglobin independently, partly offset through independent myoglobin absence across cold tissues. Different lineages reach similar physiological outcomes through distinct genetic changes, partly offset through convergent cardiovascular outcomes across cold tissues. Elevated nitric oxide signaling may help promote wide vessels and vascular growth, tied closely to nitric oxide signaling.
Nitric oxide regulates both blood flow and formation of new vessels, compensated by angiogenic vessel growth inside Antarctic tissues.
Long-term signaling can reshape anatomy throughout growth. Through developmental compensation,
vascular differences emerge as the animal develops, through heart growth trajectories, limiting stress while sustaining heart growth trajectories in Antarctica.
Static signals may transform a blood defect into a stable body plan, sustaining homeostatic circulatory remodeling throughout Antarctic activity. Low viscosity makes large volume pumping mechanically easier, partly offset through viscosity-enabled high flow across cold tissues.
Tips. Cardiovascular compensation still requires substantial energy. Through energetic compensation costs, limiting
the fish pays for weak blood with a larger circulatory apparatus, revealing large circulatory investment under stable cold conditions. Warmer water can push that enlarged system toward its performance ceiling, sustaining thermal cardiac limits throughout Antarctic activity. Cardiac capacity may limit acute warming tolerance in some species, partly offset through cardiac performance ceiling across cold tissues. Every compensation traces back to an ancient loss recorded in the genome, with consequences for integrated polar physiology. Their closest red-blooded relatives still retain that ancestral system, partly offset through retained relative physiology across cold tissues. A major deletion destroyed essential hemoglobin coating material, partly offset through beta-globin deletion across cold tissues. Additional remnants preserve a molecular fossil of the lost pathway, compensated by alpha-globin genomic fossil inside Antarctic tissues. Once a protein was no longer required, disabling mutations accumulated more freely, central to post-loss mutation accumulation. Blood physiology changed far beyond the globin genes themselves, partly offset through reduced iron physiology across cold tissues. White blood contains dramatically less iron bound in respiratory pigment, compensated by iron-poor white blood inside Antarctic tissues. The absence of red cells lowers viscosity substantially, partly offset through erythrocyte-free viscosity across cold tissues. Lower viscosity may have softened the damage without making hemoglobin loss beneficial, sustaining evolutionary trade-off interpretation throughout Antarctic activity. Environmental conditions created a rare window in which the mutation could persist, sustaining cold ancestral tolerance throughout Antarctic activity. Similar gene loss would be far less tolerable in warmer, oxygen-poor seas, which affects warm water incompatibility. Red-blooded relatives often retain greater capacity for oxygen transport during thermal stress, which affects relative thermal capacity. Extreme specialization can increase success in stable environments while reducing resilience to rapid change, supporting specialization. Genome studies reveal additional adaptations beyond blood and circulation, partly offset through antioxidant gene expansion across cold tissues. Oxygen-rich water and abundant mitochondria can increase oxidative stress, partly offset through mitochondrial oxidative pressure across cold tissues. Strong antioxidant and repair systems limit damage from reactive molecules, partly offset through macromolecular repair defense across cold tissues. Icefish survival depends on a broader Antarctic molecular toolkit, partly offset through broader cold adaptation across cold tissues. Traits optimized for constant cold may become liabilities during warming, partly offset through warming-created mismatch across cold tissues.
Climate change modifies several ecological pressures simultaneously through multi-factor climate pressure.
Risk will vary among species, locations, depths, and life stages, partly offset through species-specific exposure across cold tissues. Warm water holds less dissolved oxygen while animal metabolism accelerates, partly offset through warming oxygen decline across cold tissues. Icefish face simultaneous reductions in supply and increases in demand, compensated by aerobic supply-demand compression inside Antarctic tissues.
Limited oxygen carrying capacity may bring some ice fishes to their ceilings sooner, tied closely to earlier performance ceilings.
Thermal vulnerability is not identical across the family, partly offset through variable family vulnerability across cold tissues.
Behavioral movement may reduce exposure to warming surface layers, partly offset through behavioral thermal refuge across cold tissues. Mutagens. Suitable cold refuges may not exist everywhere. Through geographic restraints, limiting stress, while
early life stages may encounter different thermal risks than adults, compensated by larval thermal exposure inside Antarctic tissues. Warming can reshape development before circulatory compensation is fully established, balanced through developmental oxygen stress during polar life. Changing prey fields can indirectly affect icefish energy budgets, partly offset through changing prey networks across cold tissues. Reduced food could make costly cardiovascular compensation harder to maintain, partly offset through cardiovascular energy deficit across cold tissues.
Field monitoring is essential for separating short experiments from population trends, with consequences for long-term population monitoring.
Collections reveal anatomy, genetics, diet, and physiological condition, partly offset through research specimen collection across cold tissues.
Modern tools connect whole animal performance with molecular mechanisms, partly offset through integrated molecular analysis across cold tissues.
Comparative studies identify which traits followed hemoglobin loss and which evolved earlier, with consequences for comparative evolutionary mapping. Variation within the family turns icefishes into a natural evolutionary experiment, with consequences for within-family physiological variation.
Their biology offers insights into oxygen transport without providing a direct model for human treatment, supporting biomedical research boundaries.
Genes. Understanding compensation may illuminate general principles of cardiovascular plasticity, partly offset.
Those adaptations remain inseparable from the cold, oxygen-rich sea that made them possible, helping explain Southern Ocean dependence.
The future depends on whether physiology can keep pace with changing Antarctic water, tied closely to future warming pressure.
Antarctic icefish survive without red blood by rebuilding nearly every other part of oxygen delivery, supporting integrated oxygen delivery. The Great Pyramid still contains spaces whose purpose and exact shape remain unknown.
Khufu's monument belongs to a much larger royal landscape at Giza. Originally, polished limestone covered the stepped core visible today. Erosion and stone removal changed the monument's outer profile. Its builders established an exceptionally regular base.
The pyramid's orientation closely follows the cardinal directions.
Ancient surveyors had several possible methods for finding true north. The exact alignment procedure remains debated. The monument required quarrying, transport, and positioning on a massive scale. Egyptian crews relied on familiar materials, organized with extraordinary coordination. Much of the core limestone came from local quarries beside the pyramid. Workers isolated blocks by cutting trenches around them. Levers helped separate and reposition heavy stones. Sleds distributed weight across the ground. Moistening sand can reduce the force needed to pull a sled. Water transport moved selected stone closer to the plateau. High-quality casing limestone arrived from the Tura quarries. granite for internal chambers traveled from southern Egypt. The river linked distant quarries with the building site.
The pyramid depended on transport infrastructure as much as stoneworking skill.
The known interior begins with a sloping passage entering from the north. Part of the route cuts directly through the plateau's limestone. The subterranean chamber was left incomplete.
Its intended function remains uncertain.
A concealed junction branches upward toward the pyramid's main chambers. This route was carefully built within the rising masonry.
The Grand Gallery is the pyramid's most dramatic known internal space. Its walls narrow upward through overlapping courses. Architectural details suggest specialized construction or closure procedures. No single explanation for the gallery's full role is universally accepted.
A separate passage leads to the chamber, traditionally called the Queen's Chamber.
No queen was found there, and the traditional name may mislead.
The niche's original purpose is unknown. Small channels extend upward from the chamber. Exploration found doors or blocking stones within the shafts.
Copper-like fittings increased curiosity without revealing the space beyond.
Robot probes exposed another confined section, not a large treasure chamber.
The upper route passes through a complex granite antechamber. Granite barriers could seal the final chamber route. The king's chamber contains the monument's surviving stone sarcophagus.
Above the chamber, stacked spaces redistribute enormous masonry loads.
The upper roof channels pressure away from the chamber ceiling. Internal geometry was engineered to manage crushing forces.
Modern scans revealed a major unknown space above the Grand Gallery.
Its precise shape cannot yet be reconstructed. The detected zone extends for at least about 30 meters.
Muon data do not yet determine its orientation or internal divisions.
Its existence is better established than its architecture. Independent muon measurements confirmed the anomaly. Cosmic particles made the hidden space visible without drilling. Another scan detected a corridor behind the pyramid's north face chevrons.
The hidden corridor extends inward for roughly 9 meters.
Its destination and function remain uncertain. A miniature camera provided the first direct images inside.
The corridor has a gabled ceiling beneath the external chevrons. One possibility is that it protects masonry below from weight. Structural and access-related functions are both discussed. No confirmed doorway has been found at its end. Additional methods have reinforced the corridor's location. Multiple techniques now describe the same hidden structure. No proven passage currently connects the two discoveries. The pyramid's interior mixes documented corridors with unresolved anomalies. Different discoveries carry different levels of certainty. Scientists distinguish physical entry from remote imaging. Voids transmit more cosmic muons than dense masonry. Large datasets convert particle counts into internal density images. Repeated measurements reduce the chance of a false signal. The big void remained visible across independent analyses. No evidence shows that the void contains treasure or a royal burial. Its role may relate to construction, load management, or an unknown passage system. Understanding hidden spaces may reveal how the pyramid was assembled. Internal passages had to remain accessible during construction. Temporary construction routes could later disappear inside the core. No surviving plan identifies the complete ramp system. A single straight ramp becomes increasingly massive at greater height. Straight ramps remain plausible for early construction stages. Switchback ramps reduce overall ramp length. External spirals could obstruct surveying of corners and faces. Internal ramp models place some hauling routes within the structure. Archaeological confirmation for a complete internal spiral remains limited. Levers may have raised blocks through shorter vertical steps. Temporary supports could hold stones between lifts. Ropes coordinated pulling, steering, and braking. Simple direction-changing devices may have multiplied crew control. Builders probably used different methods at different stages. Material size and route requirements changed with height. Some internal granite elements demanded specialized handling. The exact lifting sequence for massive granite remains debated. Construction traces constrain theories even when they do not settle them. No exotic technology is required by the archaeological evidence. Archaeology uncovered a substantial community supporting pyramid construction.
The settlement contained infrastructure for feeding and organizing labor.
staple foods sustained large rotating work teams. Animal remains show substantial meat provisioning.
Some workers survived serious injuries with care.
Construction depended on organized crews rather than chained masses.
Specialized roles connected quarry, river, workshop, and monument.
Mera's log records limestone transport to Khufu's construction site. The diary describes repeated deliveries rather than legendary machinery.
The project mobilized resources across the Egyptian state.
Nearby burials further identify the builders as Egyptian communities. The old image of foreign slaves building the pyramid lacks archaeological support.
Labor may have mixed rotating crews with long-term skilled personnel.
The Nile cycle helped organize people and supplies.
Modern mapping has reconstructed waterways closer to pyramid sites.
Ancient channels likely shortened overland stone transport.
The pyramid complex linked river access with elevated construction zones.
Monumental routes could serve both symbolic and logistical roles. The monument was a social system as well as an engineering project.
The date and circumstances of ancient looting remain unclear.
A later tunnel bypassed the original entrance system. Modern visitors see only part of the monument's internal volume. Its architecture and purpose remain the largest interior mystery. The confirmed corridor raises a separate question about the entrance zone. Several explanations fit portions of the evidence. Non-destructive technology may refine the internal map. Better geometry could test construction and structural hypotheses. The pyramid's deepest mystery is not who built it, but why these hidden spaces exist.
A hollow tube of ice can grow downward beneath the ocean, surrounding a stream of sinking concentrated brine.
The process begins because winter air cools the sea surface far below the temperature of underlying ocean water.
When seawater freezes, water molecules join the ice lattice, while most dissolved salts remain outside the crystals.
Rejected salt concentrates the remaining liquid, producing brine that is colder and saltier than ordinary seawater during channels. Young sea ice is not a solid slab. It behaves like a porous mixture of ice and concentrated liquid. As cooling continues, connected pathways allow increasingly dense brine to drain through the sea ice under gravity. Additional dissolved salt increases density, making the rejected brine heavier than the surrounding ocean water below during ice.
Once released, the cold brine begins sinking as a narrow gravitational plume beneath the floating sea ice.
High salinity lowers the brine's freezing point, preventing the descending core from solidifying immediately. Crystallization builds growth below. The surrounding ocean can freeze when the plume cools it below the local seawater freezing temperature during sediment.
Ice, therefore, forms around the brine stream, creating the first thin wall of a hollow structure during winter. Continued discharge extends the sheath downward, Because fresh seawater freezes near the moving plume tip, freezing acts naturally. The brinacle grows from its lower edge, rather than sliding downward as one completed piece of ice. Its final shape resembles an inverted chimney, carrying a dense cold liquid downward instead of hot gas upward. Both structures organize flow within a solid wall, but temperature and gravity reverse the familiar direction during tank. A true brinacle is hollow, with an icy shell continuously surrounding a liquid brine core. Gravity acts seasonally. Brine flow controls how quickly the tube lengthens and how thick its walls become during formation, during plume. Too little flow cannot sustain growth, while excessive flow may destabilize the forming tube and surrounding water.
Stable brinicles require a narrow combination of cold discharge, favorable salinity, and relatively gentle surrounding currents during ice. The visible ice finger is the successful outcome of many hidden processes occurring within the sea ice above. Brine rejection begins at the microscopic boundary where ordered ice excludes most impurities from its structure during seafloor. Cooling reduces liquid volume and forces salts into increasingly concentrated brine inclusions within the ice. Dilution acts efficiently. Brine chemistry evolves during cooling because individual salts have different solubilities and precipitation temperatures. Flow stabilizes branching below.
Salt composition influences freezing point, viscosity, density, and the shape of any tube that later develops during seawater.
Temperature and salinity determine whether the mushy layer remains permeable enough for gravity drainage. Discharge determines growth below. If channels lose connectivity, concentrated brine remains trapped and cannot feed a sustained downward plume. Drainage acts structurally. Small thermal changes can reopen pathways and release stored brine in sudden drainage events. Pressure alters stability below. Converging drainage concentrates enough cold brine at one point to support a visible brinacle. Brine maintains persistence below. The outlet geometry helps anchor the tube while the plume begins freezing nearby seawater. Crystallization builds transport below. A reinforced upper connection supports the growing structure against its own weight and weak surrounding currents during Antarctica. Brinacle growth continues only while the overlying ice supplies sufficiently cold, concentrated brine through connected channels during ice.
When the brine source weakens, the lower growth front slows and eventually stops extending. Freezing records freezing below. An inactive tube may flood, fracture, or melt after its internal brine circulation ends. Salinity shapes exchange below. Ambient flow can bend fragile brinicles or detach them before they reach the seafloor. Transport connects flow below.
Many short-lived tubes probably disappear without ever being observed by cameras or divers. Heat flow governs formation below. Low turbulence allows a delicate ice wall to survive long enough for continued downward growth. Gravity acts repeatedly.
Crossflow tilts the descending brine and forces the ice tube to grow along a curved trajectory during interface.
Natural brinicles can twist, branch, or lean because real polar seawater is never completely motionless. Turbulence acts continuously.
Changes in plume structure can create multiple outlets and complex frozen geometries beneath the same source during tank.
Despite irregular exteriors, the defining feature remains a liquid pathway enclosed by newly frozen seawater. Cooling acts thermally.
The brinacle is a tube, not a solid spike, and its interior remains actively connected to the sea ice. Heat flows toward the supercold core, allowing seawater beside the plume to release latent heat and freeze. Every new layer of ice releases latent heat that must be carried away by the surrounding water and brine. Growth rate depends on whether nearby water can remove that released heat efficiently. Density steadily drives persistence below. Warmer surroundings or weak heat removal reduce freezing and produce thinner, less stable structures. Discharge determines transport below. Extremely favorable thermal conditions can freeze a wall quickly before the plume travels far downward. Drainage acts seasonally.
Wall growth can eventually choke its own supply, creating a self-limiting feedback within the tube during winter. Continued flow helps preserve the hollow center by delivering saltier liquid with a lower freezing point during seawater.
The lower opening must remain unsealed so the dense brine can continue sinking beyond the ice wall. The frozen tube records the path of a cold descending wake that continues beneath its visible endpoint.
Entrainment dilutes the plume, reducing density, salinity, and the ability to freeze additional water. Mixing weakens formation below. Only the colder concentrated case remains below the surrounding seawater's local freezing threshold. Freezing steadily records geometry below. Combined thermal and salt conditions decide whether the plume sinks, mixes, freezes nearby water, or disperses into the ocean.
Freezing around the plume can concentrate its remaining liquid further, reinforcing density and downward motion. Transport acts gradually.
Brinacle formation participates in a small local circulation driven by both temperature and salinity differences. Heat flow acts thermally. Surrounding seawater continually replaces liquid frozen into the expanding ice wall. Gravity steadily guides local structure below. Sediment and plankton can make local flow patterns visible around an actively growing brinacle. Chemistry modifies growth below.
The most extreme temperature and salinity conditions remain concentrated near the liquid core and lower outlet during sediment.
Brinicles range from small, fragile tubes to structures capable of extending several meters beneath sea ice during winter.
A sufficiently persistent source can carry the frozen chimney all the way toward the ocean bottom during seawater. When a brinacle reaches bottom, its dense liquid core continues flowing outward across the seafloor. Ice growth acts seasonally.
The cold brine spreads as a gravity current because it remains denser than the water above it.
Bottom topography guides the plume into channels, hollows, and low points across the polar seabed. Flow acts chemically. Nearby seawater can freeze along the spreading plume when local temperature falls below its freezing point during channels. Horizontal freezing may extend beyond the brinacle base, while the dense liquid continues moving inside and beneath it. Slow benthic animals may struggle to leave a rapidly cooling patch when the plume reaches their habitat.
Biological outcomes depend on mobility, exposure time, body size, and the precise path of the dense flow. Documented imagery can appear dramatic, but a brinacle does not instantly freeze every organism it encounters during shell.
Organisms may face freezing, osmotic stress, or simple displacement, depending on their location relative to the plume.
Hypersaline brine draws water from tissues and can challenge animals even where new ice never touches them. Sea ice brines also contain microorganisms adapted to cold, salt, and rapidly changing chemical conditions. Drainage can transport dissolved chemicals and microscopic life from the ice into the ocean below. Freezing acts naturally. A brinacle is therefore part of a broader exchange pathway between sea ice and seawater. Salinity acts briefly. Dilution eventually weakens the extreme salinity and temperature that sustain bottom freezing. Transport steadily connects local mixing below.
After discharge declines, the remaining chimney may persist briefly as an inactive frozen structure. Heat flow governs stability below.
Ocean between floating ice and fixed seafloor can bend or break a grounded brinacle. Gravity guides persistence below. Detached pieces disappear when surrounding conditions no longer support their unusually cold frozen walls. Chemistry modifies transport below.
Brinacle formation is episodic, following where drainage channels open and kloof beneath evolving winter ice. Turbulence acts chemically.
The phenomenon is most likely during active sea ice growth, when strong cooling drives concentrated brine rejection. To isolate these interacting controls, researchers recreated brinacle growth inside carefully monitored laboratory tanks. Cooling controls freezing below. Laboratory experiments reproduce brinicles by injecting dense, cold brine into water with a higher freezing point during seafloor.
The artificial tube confirms that a descending plume alone can organize the essential hollow structure. Dilution acts efficiently.
Sensors reveal how temperature gradients evolve as the wall grows and the plume entrains surrounding water during winter. Measurements distinguish concentrated brine, newly frozen water, and unchanged ambient liquid outside the tube. Density acts gradually. Adjusting discharge rate directly changes tube length, diameter, stability, and the speed of downward growth. Discharge acts thermally.
Weak flow cannot maintain the thermal plume needed to keep extending the hollow ice front. Drainage acts structurally.
Control crossflow demonstrates why natural currents can determine whether a brinacle survives or collapses. Pressure alters branching below. Different salts produce distinct growth rates, wall textures, and branching patterns under otherwise similar conditions. Brine acts briefly. Brinacle shape contains information about the chemistry and dynamics of the liquid flowing inside it. Crystallization acts steadily. Despite varied shapes, all successful laboratory examples follow the same basic thermodynamic and gravitational process. Geometry acts locally.
Brinacles resemble chemical gardens because a flowing solution builds a solid tubular wall around itself. Mixing acts seasonally.
Gravity, density, and temperature reverse the direction while preserving the self-organized hollow geometry. Freezing records persistence below.
One system deposits minerals, whereas the polar version freezes surrounding water into an icy membrane. Salinity acts chemically.
Sustained internal flow allows both structures to extend far beyond the point where formation originally began, during sediment.
Brinacle walls are active boundaries where heat transfer, salt exclusion, and fluid motion remain tightly coupled during winter. The frozen shell is not perfectly pure or impermeable, especially during rapid laboratory growth. Gravity guides freezing below. Local wall defects can redirect brine and generate branching or double-walled morphologies. Chemistry steadily modifies exchange below.
Competing channels may merge when their thermal fields overlap within the same surrounding water. Turbulence limits flow below.
Computer models test how coupled fluid dynamics and freezing reproduce the laboratory structures. Convection steadily redistributes formation below. The same physical